actually give us to work with. The first one being that the tumor tissue often yields variable DNA quality and quantity from tissues that are used for routine diagnosis. So in cancer, certainly in cancer diagnosis, cancer biopsies or surgical samples are excised. They're then fixed with formalin and then embedded in a paraffin wax cube or paraffin wax cassette. And this is because that type of reagent is very easy for sectioning and looking at cells down the microscope. Unfortunately, it's absolutely terrible for DNA, just illustrated by this slide here. So this is a DNA ladder. These are 12 samples all taken from the same cancer type from the same hospital. One sample gave nice high molecular weight DNA, much higher than the ladder. You can see as you move from left to right, you get increasingly worse DNA. This is actually highly fragmented. Luckily, Illumina sequencing is relatively short sequencing reads, so about 50 base pairs at the low end to 300 at the high end. So even these small fragments are still useful for DNA sequencing, but there is this additional challenge of just making this DNA sequenceable or compatible with the Illumina sequencers. The other challenge is that tumors are not just perfectly homogeneous masses of cancer cells. They're actually a mixture of tumor cells and invading or surrounding normal cells. So this is actually a screenshot from a laser capture microdissection experiment. This is a lung cancer that's metastasized to a lymph node, so it's already very rich in lymph node cells, immune cells. What we've done here is actually circled all the tumor deposits, and in this case, to enrich the number of cancer cells for analysis, we use the laser to cut out these cells and capture those, and then do DNA extraction from purified cells. And sequencing has gotten cheaper and cheaper. A secondary approach has arisen where we'll just sequence all of this DNA together and sequence this actually to great depth instead. So instead of just generating 15 reads like I showed in that germline example, we'll generate 150 or 1,500 or 15,000. Just sequence past normal in the search for somatic mutations in highly impure tumors. So you may hear this concept of contaminating stromal cells referred to as tumor purity or also tumor content. So the two buzzwords that you'll see to describe this idea of tumors being a mixture of cancer cells and non-cancer cells. The other challenge I touched on earlier is tumors can have multiple copies of chromosomes. So that assumption of 2 copies of every gene is broken in cancer, and even more confounding, not every chromosome is necessarily at the same copy state. Chromosome 1 here is a 4 copy, chromosome 2 is a 3 copy. It'll actually be easier to find mutations potentially in the 3 copy chromosomes versus the 4 copy chromosomes. So you may hear this concept referred to as aneuploidy or ploidy for short. One way around this is to sequence the DNA from these admixed tumors to death. This is where exome sequencing and certainly targeted sequencing still really has a home. This is the same sample sequenced 2 ways, genome sequencing and exome sequencing. In this case, this region of the genome has only 15 reads. The exome has nearly 140, and that really gave the algorithm or the alignment the power to pick up the mutations that actually, or the DNA fragments that correspond to the reads supporting this actual mutation. So really it's trying to think about what is the goal of the paradigmatic analysis. Is it to find absolutely every mutation in a comprehensive way, sequencing very, very deeply, or is it a survey of cancer genome variation to acknowledge that in low coverage regions, especially in highly aneuploid or low purity tumors, mutations may be missed. One benefit of the deep sequencing approach is the ability to infer tumor purity, ploidy, and potentially subclonal structure as well if you have the depth to detect these very rare variants. This is one algorithm we use in my lab called Sequenza. The way to read this plot here, the chromosomes ordered from left to right. Each dot here is a variant, a germline variant, or some sort of difference in the genome. And you can see just by eye here sort of these copy number changes as you sort of get these shifts in both allele coverage, but also allele-specific coverage, but also in coverage across the whole genome itself. The goal being to generate these genome-wide copy number alteration plots, where you should have 1 copy of red allele, 1 copy of a blue allele. So a normal cell here would just have 2 blue and red lines that just line up nicely along this baseline. In this case, this is a highly aneuploid tumor. Lots of copy number gains, lots of deletions. Actually, this chromosome here is quite interesting, where one allele was just deleted, so that is basically a copy 0. And to stay alive, the cell actually duplicated the other chromosome. So it still has 2 chromosomes, they're just both the same. These algorithms can infer both tumor content and average ploidy. These are inferred from knowledge that normal cells can contribute DNA as well. So you're not necessarily expecting these perfect 50-50 balances between mutant and non-mutant allele. You actually get this sort of dilution effect of having normal cell contamination. And then tools like PyClone PhyloWGS is another tool that will take this type of data, they'll take your somatic mutations, and then basically answer the question or give you a readout into what percentage of the cancer cells have these mutations. So in this case, all of these mutations here are all in 100% of the cells. These mutations here are in roughly 80% of the cells. And here are subclonal mutations that are often visible to shallow whole genome sequencing that are, in this case, only visible in 20% of cells. So it's something that you can see with deeper profiling and can then assign to specific subclones. In this case, very important driver mutation present in the clonal cancer population. As Francis alluded to, Illumina sequencing isn't the be-all and end-all of DNA sequencing in the world. I've just shown an example here where we use long-read PacBio sequencing to complement what we have from Illumina sequencing. So in this case, we discovered mutations in these 2 genes here. We wanted to confirm that those mutations were real. So in this case, this is a point mutation, so the base changed colors, so you can see it. This was an out-of-frame 2-base deletion. And we wanted to ask the question, can we see those in PacBio data as well? The real challenge with this technology, at least at the time, this is a little old now, you can see the real variant shining through, but you can see this background error noise. What's useful is the background errors are not necessarily always in the same place, but they're certainly there, so you also have this background effect that can make variant discovery sort of more challenging than it would be in cleaner Illumina data. Certainly not really a problem for point mutations, because the reads that have the variants are very clear. This we thought might be more of a challenge in the indel space, because the types of errors that the PacBio sequencer makes were specifically insertions and deletions. But you can see this variant was very highly recurrent, so something that we could call out on both platforms. This is where it's very useful, especially if you're looking for challenging types of cancer genome variation, to have an orthogonal technology to validate what was discovered in one platform with something that's more complementary. I want to talk a little bit about depth, why sequence deeper. In addition to the overcoming the challenges of purity and ploidy, the ability to really detect very, very rare sequencing variants. I've shown the plot of how low can you go in terms of allele fraction and with depth. We actually just published this last year in a ctDNA study. Exome sequencing is sort of being in this realm of 100 to 200x coverage. The clinical lab will sequence our panels to 500x plus. If you really want to get serious about subclonality, we'll use highly targeted custom assays to look for these very rare subclones. And circulating tumor DNA, we're currently sequencing those to about 25,000x coverage. In this case, this is DNA from tumors that have been shed into the bloodstream, so we have to sequence past all the contaminating normal DNA fragments and find the 5, 10, 15 fragments of DNA that are derived from tumors. You can't necessarily. So we'll often, certainly in that study, we had the tumor, we had a bone marrow aspirate in the case of multiple myeloma. Actually, I'll just jump here. So we knew what the answer key was. We knew where the somatic mutations were. But we did have several examples where we found mutations, definitely drivers we'd seen before, that weren't in the primary tumor. Multiple myeloma is multiple right in the name. You have many of these masses, and I'm convinced those mutations must have been in some other mass in the body. But you couldn't necessarily say, we found this in the blood, it's that one versus that one. It's present, but you don't necessarily assign them to a certain tumor mass. So here's sort of a now famous cartoon of where circulating tumor DNA can come from. So I do want to differentiate cell-free DNA from circulating tumor cells. These are 2 sort of flavors of a similar idea. I'm going to talk specifically about cell-free DNA. So this is DNA that comes from tumor cells that are turning over, dying, shedding their DNA into the bloodstream, and is now dissolved in the plasma portion of the blood. There are also circulating tumor cells. They're much harder to find, often at lower concentrations. These are actually in the cellular portion specifically of blood. I'm going to be talking about the liquid portion, which is a combination of 99+% normal cells and 1% less cancer cells. Here's what next-gen sequencing data looks like from that type of data. It's from the two papers, or two technologies that were published. One's called TAMseq. This is a PCR-based approach. You put your primers down, you amplify only your region of interest. You can see highly focused. Here are two exons down here. They really wanted to find basically mutations only in these exons. This is a little more challenging in that it requires highly multiplexed polymerase chain reaction, so highly multiplexed PCR. The other challenge here is it's very hard to scale this into larger, especially contiguous regions of the genome. So another approach using hybrid captors is the same technology used for exome sequencing. You put your bait down 2 exons here. This will actually pull down sort of regions of DNA. This is the method we certainly use in my lab to tile across regions that are very difficult to walk across with PCR reactions, but something you can just put in probes, not just on exons of interest, but really in any arbitrary region of interest. It's conceptually exactly the same as exome sequencing. Rather, you're just focusing the panel design much more and sequencing to much greater depth.